right, part 2 of exercise 4 here. We're going to concentrate on actual staining. I'm going to talk a little bit about staining in general first, and then more specifically about the gram stain. Okay, so I'll come back to this first picture because this is hopefully what your gram stain will look like today. All right, little staining theory here. There's two kinds of stain, kind of broad categories. Basic stains and acidic stains. A basic stain or a basic dye, that just means the colored portion of the stain carries a net positive charge. Okay, and that's important because cell walls, what kind of charge do they have? They have a slight negative charge. Okay, so unlike charges attract, when the basic stain has a net positive charge, that's going to the positive charge of the stain is going to adhere very well to that negatively charged cell wall. Now, there are other parts of the cell that have a slight negative charge, like DNA, some proteins maybe, but of course it's going to come to the cell wall first. Not the cell membrane. Remember, the cell wall is on the outside. Okay, so some examples of basic stain that we're going to use today: crystal violet, safranin. Most of them are going to be basic dyes. Manivals. I don't usually do all these things in the same day, but okay. Acidic stains, then, as you may have guessed, is the opposite. So the colored portion of the stain carries a net negative charge. Okay, so it's going to be repelled by the bacterial cell wall. And all bacterial cell walls have a net negative charge. Okay, so because it's repelled by the bacterial cell wall, so acidic stains are going to stain the background instead of the cell itself. Either way, you get good contrast, whether you stain the cell and not the background, or you stain the background and not the cell. You're going to get good contrast. Okay, and we call this negative staining. Okay, so our example of an acidic stain that we're going to be using today when we do the capsule stain is called Congo Red. Okay, and we use that strategy to visualize capsules because it's very difficult to stain the capsule itself. So we basically stain everything else and then we can see the capsule. All right, two other categories when we're thinking about staining in general. If we're doing simple staining, that means that we're going to use one dye or one stain to see the cells. And a lot of introductory microbiology labs do this. You get your cells on the slide, you add one stain, a couple minutes, you rinse it off, and you take a look under the microscope. And that can be helpful if you just want to see the cell shape, or we could say morphology. You know, is it a rod-shaped cell? Is it a cockeye? Okay, that's going to give us that information if you want to tell how big the cell is, the size, or also the cell arrangement. So that helps us identify organisms as well. Do the cells tend to cluster together, for example, or do they tend to form chains? Okay, that can be a big tool that tells us something about the cell. But beyond that, I mentioned the size. That's pretty much all the information we would get out of simple staining. all the staining procedures that we're going to do in this class are considered differential staining procedures because they're going to use 2 or more dyes in order to classify or put the cells into groups. Certain cells will be one color, certain cells another, or maybe to visualize a particular structure. Okay, so for example, today we're going to be doing gram staining, and that will help us understand if the organisms are what we call gram positive or gram negative. Okay, that doesn't have anything to do with the charge of the cell, it's just positive for the gram reaction or negative for the gram reaction. Okay, gram positive stain purple, gram negative stain pink. The capsule stain that we're also going to do today, we use a couple different dyes and it helps us to see a particular structure, the bacterial capsule. Now, if we have time after Thanksgiving and we're still meeting in person, we will do an acid-fast stain as well as a spore stain. Okay, acid-fast, that's a category of organisms with a specific kind of cell wall that's not really gram-positive or gram-negative. So we're using a couple different dyes to see if the cell is in that acid-fast group. A spore stain, again, that's another particular structure. So I lobbied hard for us to find time to do those. I didn't win. We're going to do that after Thanksgiving again if we are still meeting in person. Okay, let's talk specifically about the gram stain. This is definitely the most important staining procedure that you will learn in this class. Okay, what's the difference between an organism that's considered gram positive or gram negative? That's what we really want to understand to begin with here. So if we take the cross section of a cell that's considered gram positive or positive for the gram reaction. Again, if we took a cross section of the cell, of course, here's the cell membrane on the inside. Outside of that, if we consider this part the cell wall, it's really not too diverse. Okay, about 90% by weight of that cell wall of a gram-positive cell is one compound, and hopefully you talk about this a lot more in lecture. It's called peptidoglycan. Now there's some other things in there, teichoic acids, lipoteichoic acids, but the main thing there is peptidoglycan. And if you what is peptidoglycan? Well, when you see this portion of the word peptid, that should make you think peptide. Okay, there's some amino acid cross bridges in that compound. Glycan, that part of the word should make you think sugar. Okay, so there's a couple of different sugars, alternating sugars, kind of very similar in structure to glucose, and then they're kind of cross-linked with these amino acid bridges. So it's a very rigid structure, and again, gram-positive cells have a whole lot of it, and that really makes them hold on to dyes for a lot longer instead of being very easily decolorized. Okay, the gram-negative is a little bit more complicated cross-section. So if we take, here's the cell membrane, outside of that, we have really a very thin amount of peptidoglycan. By weight, it's only about 10%. Okay, and then outside of that, there's an outer membrane for gram negatives. It's got some proteins in there, a compound called LPS, lipopolysaccharide. Okay, so the main thing here in terms of what color the cells stain is that because there's so little peptidoglycan, again, it gets destained very quickly. Okay, so a mnemonic that helps me kind of remember, maybe it will help you, peptidoglycan or gram positives, again, have a lot of peptidoglycan, very thick, large amount in their cell wall. Gram negatives only have a little bit, so kind of a little thinner amount, again, get destained a lot easier. Okay, so I'm going to be demonstrating in class how to do the gram stain. Basically, we are going to be doing 2 gram stains, I believe. In one, we're going to take cells from a broth, and in one, we're going to take cells from a plate. Okay, so when you take cells from a broth to make up your bacterial smear, by the way, first of all, I would suggest that you always label the one end of your slide with maybe, say, your initials or whatever, just so you don't get it mixed up with anybody else's. But more importantly, so you know which side of the cell you've put your cells on, because it's easy enough to drop the cells while you drop the slide while you're staining it. So I know that I've put it on the side where I can read my initials there. Okay, so when you're putting your cells from a broth, I would take 1 or 2 loopfuls. Put 1 loopful on here on your slide, kind of spread it around, maybe dime or nickel size. Flame your loop, go back, grab another loopful, and then mix another loopful right on the same place. You always have less cells when you're taking cells from a broth, so it doesn't hurt to take a couple loopfuls. But again, you want to clean your loop off. You don't want to take the chance that you're going to contaminate that stock culture. Okay. The difference is when you take cells from a plate to make up your bacterial smear, you're going to add a drop of water and put that on the slide first. You want to get kind of the smallest drop that you possibly can. I'll give you some hints how to do that. And then, since you're taking from a plate, it's best to use your needle when you get your cells from a plate. Basically, that's just a loop without the little loop at the end. Just touch some cells and then mix it in with that drop of water. Okay, the reason is that if you use a loop, you tend to get way more cells than what you need, and they kind of clump together and it's hard to see their actual shape. So if you use a needle, again, just touch a colony and you'll have a whole bunch of cells in there. Okay, regardless of whether you took from a broth or a plate though, you always are going to need to heat fix before you start your staining procedure. And you can do that either by passing it through the Bunsen burner, again, which I'll demonstrate, or you put it on the hot plate on low until it's totally dry. If you don't heat fix, then a lot of the cells are just going to slide right off when we start our staining procedure. I believe the book says that you need to air dry before you heat fix. I have found that that's not really necessary to do that, that the cells aren't distorted at all by not air drying them. It just takes longer when you have to let them sit there and air dry. So I wouldn't worry about that. Personally, I don't think it's really necessary. All right, so I'll show you how to heat fix. And then when you start your staining, you've got a really nice diagram on page 20, I believe it is, in your book. So you're going to start out with the crystal violet and just put that right on your stain there. That's called our primary. We'll call it I'll write it this way. Oh, that's not readable. Primary stain, crystal violet, that's, remember, a basic stain. It's going to adhere to the cell walls. You're going to leave that on there for a minute, rinse it off, and then you're going to add grams iodine. Okay, you're going to leave that on for a minute. This is called a Mordant Step. A mordant is something that forms an insoluble complex with something else. So basically the iodine forms a complex with that crystal violet and really hammers it into the cell wall. The next step is probably the most difficult, the one you need to pay most attention to. That's a decolorization step. You're going to add acetone that's dissolved in alcohol. Again, that's a decolorizer. You're only going to leave that on for a few seconds. So like maybe 3 seconds. Maybe a little bit if you thought you had a ton of cells on there. If you're taking, then you're just going to do it for a few seconds. Rinse it off. Right. So now after this step, your gram-positives will still have hold of that primary stain and they'll be purple, but your gram-negatives will be colorless. So we still want to be able to see the gram-negatives, so we're going to flood your smear with safranin. That's another basic dye. It's going to stick to the cell walls. Leave that on there for a minute, and now your gram-negatives will stain that red from the safranin, and the gram-positives will still be the purple of that crystal violet. We're always going to want to make sure our slide is dry before we plop it on the microscope stage, so I will show you how to blot that dry, and then you're all set. So what are you looking for? I'll go back to the beginning here. Our gram-positives, I think they're staph epidermidis. Is what is in the mix. They're going to be these purple round cocci. Cocci is our name for round cells. The E. coli are the gram-negatives, and they're going to be these pink bacilli, these pink rod-shaped cells. So a good gram stain has very distinctly purple and distinctly pink cells. If all your cells are kind of purpley pink, then either you probably you decolorize too long. Okay, so and hopefully you won't see too much of this clumping. If you get too many cells on there, then they all clump together and it's, again, hard to see their morphology. But usually you can focus somewhere where they're kind of off by themselves. All right, next slide, we'll talk about capsule staining.